So far we have been talking about dyeing and its procedure, the basics of dyeing, the typical natural dyeing recipes and so on and so forth. Today's lecture is dedicated to the preparation for dyeing, preparation of the cloth, how the cloth should be prepared before it is taken for dyeing. We know that there are the simplest way of preparing the cloth is to wash it, but there are certain very specific procedures that need to be followed when dyeing has to be done, be it dyeing with synthetic dyes or be it dyeing with natural dyes. One needs to prepare the cloth very effectively for a better dye uptake. Now, the methodology of preparation of cloth is the following. Grey cloth, as it comes from the loom stage, is unattractive and contains natural as well as added impurities, which hinders the successful operations of dyeing by reducing the absorbency of the fabric. That is why it is necessary to make the fabric water absorbent by making the fabric free from any natural as well as added impurities in order to achieve successful dyeing process. So, one thing should be very clear, that when the grey cloth is coming after the weaving of the cloth, it is to be remembered that it needs to be purified, and this purification is in terms of its washing. And because that will, this washing of the waxes, of the oils, of the impurities will enhance the absorbency of the fabric. We know that if the water absorbency enhances, the dye uptake automatically enhances in the case of cellulosic fiber as well as in the case of proteinaceous fiber. So, whether it is cotton or whether it is wool or silk, the first thing that one needs to do with the grey cloth is the raw, unwashed, unbleached cloth is called grey cloth. It contains lot of natural impurities and some of them are even added impurities. So, these should be removed before it is prepared for dyeing. Preparation of cotton cloth contains following steps systematically. In order to prepare cotton, which is toughest to dye, we have to take lot of precautions, because unless and until the cotton is washed properly, it will not have good absorbency, and if it does not have good absorbency because of the waxes and the oils on the cotton surface, it is going to be even tough to dye it. So, let us try to look at the procedure. Different treatments to the cotton fabric are: it needs to be desized, it needs to be scoured, and it needs to be bleached finally. So, there are three main steps that cotton needs to undergo. One is desizing, the other one is scouring, and the third one is bleaching. Desizing, let us try to understand what exactly is meant by desizing. Desizing is a process by which fatty matters are removed from the grey cloth, such as starch, which applied to the warp and weft yarn during weaving in order to withstand the stress or strain. See what happens, cotton is very tender. So, in order to draw it into yarns, and then the yarns being woven in the warp and the weft, what happens is that this starch has to be added. If starch is not added, cotton is very soft and the yarn would break during the process of weaving. Now, in order to facilitate weaving, starch is particularly added to the yarn. The process of desizing can be done either by hydrolytic desizing or by oxidative desizing. So, desizing or removal of such starchy impurities is done by two methods. One is the hydrolytic method and the other one is the oxidative method. Either of them are used. It is not that both the processes are used. The theme of desizing process is only to convert insoluble starch into soluble form. So, because this insolubility will further create a non-absorbency of dye solution at the later stage of dyeing, that is why it is important to remove starch. What if the starch is not removed? If starch is not removed from the fabric, it will only create more dye aggregates to settle in one place and it will affect the dye absorbency. I told you, if you recall, that by capillary action between the mesh there are pores. When the warp and the weft, these are two dimensional threads that weave the cloth. When they are woven, there are small pores. Now, it is through these pores, by the capillary action, the dye enters the fabric, and that is what we call as dye uptake. Now, dye uptake will be hindered if starch is present, because starch is in the insoluble form. So, this insoluble starch needs to be converted into the soluble form, and that is the dextrin. And the dextrin is then further converted into maltose, and the maltose is converted into glucose. So, from one form to the other, the whole idea of this conversion from starch to glucose by the hydrolytic process or by the oxidative process is to convert insoluble to soluble form. Now, similarly, the next step that is done to the fabric, cotton fabric particularly, but this is also true for silk and wool, is the scouring step. So, only desizing is done in the case of cotton, but scouring is common to cotton and silk and wool all. Scouring is a process by which oils, fats, waxes and other nitrogenous matters are removed. The process is carried out by adding 2 gram per liter caustic soda or 1 gram per liter soda ash or 1 gram per liter TRO, that is the terpene oil, and then the temperature is raised to boiling, and the process continued for 3 to 4 hours under the pH of 10 to 11.5. That means, under alkaline condition, the whole substance fabric, be it cotton, be it silk, be it wool, are washed in order to remove the oils and fats and waxes. Now, TRO is a solvent, and therefore, it is like petroleum oil, it is derived from petroleum ether, and so it solubilizes the oils and the fats and the waxes, and thus it even washes away these, and the caustic soda acts like a washing powder to remove these, but this is a very harsh scouring method. There are milder scouring methods with milder soaps also possible, particularly for silk and wool. Now, bleaching is very essential when we are dyeing, we are about to dye cotton. Bleaching is a must. A method of bleaching: the fabric is rinsed, stuck against a stone so as to remove as much of the sizing as possible. It is then spread out for sun bleaching and drying. Water is sprinkled over the cloth at short intervals until evening. This is finally washed and dried. So, this is like sun bleaching, but there are now chlorine bleaches, there are hydrogen peroxide bleaching and so on and so forth. But the traditional method of bleaching is by washing it and putting it in the sun. The UV rays of the sun actually act like a bleaching agent, and removed any kind of light colored substance which may be causing any coloration to the fabric. So, the fabric then becomes quite white and ready to be used for dyeing. Treatment of fabric before dyeing. Now, as we know that in the case of cotton, we also did a treatment of tannic acid. Time and again I am repeating this word because, or this sentence, because tannic acid treatment is only associated with cotton, and right now we are trying to see how cotton fabric is prepared. And as we are going along, I am also giving you an insight into the preparation of silk and wool. So, fleetingly I am also mentioning what is necessary for preparation of silk and wool, but primarily we are focusing on the preparation of cotton fabric for dyeing. So, after removing the impurities of fabric, it is then treated with 4% weight of the fabric solution of tannic acid in water. The fabric should be dipped in tannic acid solution for at least 4 to 5 hours. It is squeezed and dried. After mordanting, this fabric is used for dyeing, and dyeing would depend upon the type of mordanting that has to be subsequently carried on. For our study, only pre-mordanting was carried out, but of course, it is possible to do post-mordanting and simultaneous mordanting as well. So, before we go on to the mordanting, let us spend a little more time on the pre-treatment of cotton with tannic acid. This tannic acid could be from the bio sources, like one can use myrobalan, or one can use Terminalia cebula, which is myrobalan, or one can use Quercus infectoria, which is gallnut, and these two are natural tannic acid containing bio treatments, whereas one can even treat the cotton fabric with tannic acid which has been synthesized in the laboratory. So, whether we use synthetic tannic acid, or whether we use isolated tannic acid, or whether we use crude gallnut powder or myrobalan powder, all give the tannic acid, and tannic acid treatment must be done just before the mordanting, because it is not advisable to keep the tannic acid treated fabric. It will in due course of time, due to air oxidation, it creates a greenish tinge on the fabric. So, the prime reason for using tannic acid is to facilitate the dye uptake for cotton, and the second reason for using tannic acid is that it adding surfaces onto the fabric which lacks due to desizing and bleaching and all that, the three processes that we just saw, that has actually removed the attaching groups from the surface of the fabric, and therefore, tannic acid treatment provides bonding edges for the dye to come and attach. So, that is how the tannic acid plays a very important role. And after tannic acid, of course, then we have the process of mordanting. We have the choice of mordanting by 3 different methods: pre-mordanting, post-mordanting, or simultaneous mordanting, and we have studied this time and again. I have been telling about mordanting. Pre-mordanting fabric which is ready treated with tannic acid is now dipped into 4% weight of the mordant solution, not necessarily always 4%. We have seen that in the case of copper and chromium mordant, even 1 to 2% is sufficient to provide the desired shape, and therefore, we should not dose too much of metal mordant onto the fabric. Only that much percentage should be used which is required for the fabric for the adhesion of the dye. So, after that it is then squeezed and it is dried. You will see that after tannic acid treatment, or after modern treatment, the fabric is never ever washed. It is simply squeezed and dried. So, what happens, the excess of the tannic acid solution or the excess of the mordant solution just runs off, and whatever is adhering to the fabric is what is required. On both the sides, the fabric will have the mordant and the tannic acid, and that is what is the requisite amount that should be present on the fabric. No excess is required. Now, one very important innovation that we did in the laboratory was use of sonicator. So, I am just trying to take you through the important features of dyeing. We know conventional dyeing has been practiced for years, but has it made any impact by improving the process? Has the industry been benefited? No, because conventional dyes do have their own disadvantages. There are dyes where the dye gets deteriorated or discolored, or there are problems of doing it faster because of the production size and so on and so forth. So, we had developed a process of sonicator dyeing with natural dyes. For dyeing in sonicator has been a very popularized technique now. Lot of industry is interested, and there are collaborations happening where we are suggesting the use of big dye baths which have transducers fitted at the bottom and which can be used for sonicator dyeing. Extracted dye is kept in sonicator and treated fabric is dipped in for an hour. Maximum to maximum an hour is required. After 1 hour, it is dried in shade. Dye uptake of the fabric is monitored by the lowering of the optical density of the dye bath solution, and also by the shade of the color that appears on the fabric. Now, you can make out that what is meant by the shade of the color, the depth of the color, because yesterday we did that because of this color matching machine, we are able to ascertain the LAB values, the K by S values. So, this is what it means, and the UV visible spectrophotometer helps us to find out the color depth in the dye bath solution, because it is simple phenomena. See, if we make a dye bath of a certain concentration, say X concentration, and after dyeing, the dye bath will never have X concentration, it will always have X minus Y concentration. Now, this X minus Y is what has actually gone onto the fabric, because the dye cannot disappear anywhere. It has to be either in the dye bath or on the fabric that was dipped. So, this particular phenomena can be analyzed on UV visible spectrophotometer, and the color depth and its reflectance and transmittance and all that can be found out with the help of K by S, which is measured by the color scanning machine. Now, another thing that was observed, that when we do mordanting, it is stepwise process. That means, first tannic acid treatment, then the fabric is dried, then mordanting treatment, fabric is dried, and then finally dyeing. So, it involves 3 steps. Even if we take silk and wool, it is 2 step. And naturally, when there is multi-step procedure offered to the industry, it is always very cumbersome. It is tedious, because any extra step in the dyeing process can actually create waste of money, waste of time, waste of energy. So, if there is a possibility to kind of merge 1 or 2 steps, there would be saving of time, saving of cost, saving of energy. So, you see that it is going to be advantageous. So, multi-step dyeing, which was popularly the conventional method of dyeing by going stepwise, tannic acid step, mordanting step, dyeing step, change in dyeing process can result into better dye uptake. In case of BioModerns and enzyme, this was observed. So, can we now do some alteration with the metal mordant by replacing it with BioModern or enzyme? Can we merge these processes for the betterment or for the ease for the industry people to follow it? Yes, the answer is that one step dyeing process can be introduced and has been introduced. In one step dyeing process, dyeing is carried out with mordanting, that is simultaneous dyeing with the pre-treatment. So, pre-treated cloth, because tannic acid cannot be added into it. So, therefore, after the pre-treatment of the tannic acid, the cloth can be simply put into a bath where the mordant and the dye both are present. So, that would reduce the step, and from a multi-step dyeing process it can be brought down to one or step dyeing. Steps in dyeing, then there are two step dyeing also. The two step dyeing process, first mordanting is carried out by enzyme or biomodern or mordant, and then dyeing is done. Both the processes produce different results, and sometimes one step dyeing gave very good results. Now, a revived interest in the use of natural dyes in textile coloration has been growing, and there is a growing need for the availability of natural dye yielding plants. Thus, there is a strict need to explore our flora and fauna for high dye yielding plants to get new hues and shades in fabric to suit fashion conscious new age customers' demand. So, if we try to look at the whole dyeing process from the industry point of view, always it is a welcoming change that any innovation in the process, if it can reduce three things, that is the cost, the energy consumption and the time, then it is a welcoming change, and such innovations are most wholeheartedly accepted by the industry. Now, let us try to see why was it necessary to replace metal mordants. We know that all along we have been talking about the eco-friendliness of natural dyes. Now, because of the dosing or the usage of metal mordants, particularly the copper and the chromium metal mordants, which are under the category of toxic heavy metals, we had a limitation there, and because the effluent still has about 2 to 4% of these metal salts after the mordanting of the fabric, and it is run out into the river streams or water bodies or even into the agricultural land, it has its own ecological impact on this effluent not being able to recover these metal mordants. But at the same time, we know that natural dyes cannot be used without the help of these metal mordants. So, is there a method that we can somehow reduce the quantity of the metal or use a biomordant which can be a source of metal, but at the same time it has so little the metal component just optimum for the reaction or the dyeing to be sufficient for with this metal mordant present in the biomordant. Basically, even enzymes of course have a different role to play, but enzymes somehow participate in a manner which is different from the metal mordant or the biomordants which are having metal content, but they offer sites of attachment, and because they offer sites of attachment of the dyes, when they are applied on the fabric, they offer the dyes to come and sit on that. So, basically enzymes are biological catalysts. The biological means that substances in question is produced or is derived from living organism, and catalyst denotes a substance that has the ability to increase the rate of chemical reaction and is not changed or destroyed by the chemical reaction that it accelerates. Generally speaking, catalysts are specific in nature as to the type of reaction they can catalyze. So, this is the kind of general enunciation of the enzyme, general how it is considered, but enzymes as a subclass of catalysts are very specific in nature. Each enzyme can act to catalyze only very selected chemical reactions and only with very selected substance. So, the only drawback with enzymes is that they are very selective and they act more like a catalyst, and since they are from the biological origin, their reactivity is kind of just like a lock and key arrangement. Not every key can open every lock. Similarly, not every enzyme is compatible with every dye. So, one has to make a study where dye is shown to have a lock and key arrangement with the enzyme. Only then that enzyme will be effective on that particular dye. So, this kind of matching has to be made about the compatibility of the dye and the enzyme when we are using enzymes as biomoderns. Enzymes nowadays are gaining an increasing role in textile wet processing due to their proven flexibility, reliability and concern about safety, energy and water conservation and environmental responsibility. Enzyme can be used in chemical as well as biochemical processes, as they are the most efficient under normal conditions of temperature, pressure and pH. Enzymes are very specific with their action and they show results in reasonably good time and in a very cost effective manner. Enzymes of bio origin, thus making the process eco-friendly. Why enzymes were accepted? Because they are from the biological origin. So, they are like biocatalysts and therefore, they can be degraded and therefore, they are eco-friendly. Types of enzymes that are used in the textile industry: amylases, proteases, lipases, cellulases, beta glucanases or gumase, pectinase, trypsin are the common enzymes with various functions in biological systems and industrial uses, and some of which can also be proposed to use as moderns in the natural dyeing process. Among those, cellulase, protease and trypsins have been taken into study by us, and we have shown that these really work very well with the natural dyeing and the natural dyes. For example, if we try to take example of cellulases, cellulase of various types break down the complex molecule of cellulose into more digestible components of single or multi sugars. Cellulose refers to a group of enzymes which act together and hydrolyze cellulose. Cellulose is a linear polysaccharide of glucose residues connected by beta 1,4 linkages. Like chitin, it is not cross-linked. Native crystalline cellulose is insoluble and occurs as fibers of densely packed hydrogen bonded and hydroglucose chains of 15 to 10,000 glucose units. So, cellulases work on cellulose as the name suggests. So, if you remember that much, and the structure of cellulose is that it has a 1,4 linkage of the beta type of various glucose rings which are attached to one another. Its density and complexity make it very resistant to hydrolysis without preliminary chemical or mechanical degradation or swelling. In nature, cellulose is usually associated with other polysaccharides such as xylan and lignin. It is the skeleton basis of plant cell walls. cellulase enzyme have gained industrial acceptance for finishing off the cellulosic fiber or fabric to achieve a variety of effects, including enhancement of fabric surface appearance and softening of denim garments without or with low environmental impacts. Enzymatic treatments are usually performed prior or subsequent to dyeing and finishing processes. So, enzymes and the use of enzyme and the use of cellulase particularly has already been there in the textile industry, but for using it for natural dyeing, it has been used for the first time by us because it is compatible with cotton. And so, if one has to enhance the cotton dyeing property, use of cellulase really helps us, and it helps the dye uptake. Similarly, proteases are a class of enzymes referred to an enzymes whose catalytic function is to hydrolyze or break down protein, as the name suggests, proteases. So, protein. They are also called proteolytic enzymes or proteinases. Proteases can either break specific peptide bonds or, depending on the amino acid sequence of a protein, or break down a complete peptide to amino acid, that is unlimited or limited proteolases can take place. It can break down to peptide bonds only, or it can break down to its most smallest unit, that is the amino acid. The activity can be destructive change, abolishing a protein's function or digesting it to its principal components. It can be an activation of a function, or it can be a signal in a signaling pathway. However, we are concerned with proteases, how they can work in the dyeing system. The process is called proteolytic cleavage, a common mechanism of activation or inactivation of enzyme, especially involved in blood coagulation and digestion. These are where the protease is used, but when it comes to, they use a molecule of water, and thus they are also classified as hydrolases. Proteases which split up proteins into components of amino acid building blocks. Proteolytic enzymes are very important as they break down the peptide bonds in the protein to liberate the amino acids. Additionally, proteolytic enzymes have been used for a long time in various forms of therapy. So, there are hundreds and hundreds of uses of proteases, but what we are concerned or what we are interested in is the use of proteases in the hydrolysis or in the activation or in the biomodenting of wool and silk. Similarly, there is another enzyme called trypsin. Trypsin enzyme that acts to degrade protein. It is often referred to as proteolytic enzyme. It is one of the proteases or proteinaceous. Trypsin is one of the three principal digestive proteinases, and other being pepsin and chymotrypsin. But we are not concerned mainly with the digestive action of the trypsin. What we are interested that how trypsin cleaves the peptide chain mainly at the carboxyl side of the amino acid, and therefore, can it help us in the dyeing process, can it act as a biomodern, and that is what is of prime concern. Role of plants as biomoderns. A biomodern can be defined as a natural material having one or more metal ion in it, which can act as mordant. So, either we use metal mordant salt directly or use a biomodern source which has metal salt, but in a very small but requisite amount. The special thing about this material is that they are always obtained biologically, that is, they may come from vegetative matter. The presence of metal species combines with fabric in the same way as the synthetic metal salt species would do, and form the complex with dye to give stability in terms of color fastness to fabric. The research in the field of biomodern is currently going on fast pace, as this could change the picture of dyeing process in a big way. if we are able to use biomoderns, then the lot of problem that is created by the metal moderns can be overcome. Various studies have been done with plants or plant parts as potential biomoderns. Two indigenous plants, that is Pyrus pastia and Urea acuminata, which were easily available in the northeast region of India, were analyzed for their potential as biomoderns. So, two plants have been already identified by us, that is Pyrus pastia and Urea acuminata, and they have been found to have excellent biomoderning capability. Now, this is a picture of Pyrus pastia, and by the analysis of the fruit of this Pyrus pastia, which is more like berries, it was found that copper metal was present, and it was analyzed on atomic absorption spectrometer. Similarly, this is a picture of Urea acuminata. The leaves of Urea acuminata have the presence of aluminum, and this aluminum also was analyzed through atomic absorption spectroscopy. So, you see that these two plants already have, and there may be many more such plants which have metal salts in their fruit or flower or bark or leaves, and those plants can be used as biomoderns, and completely the metal moderating step can be eliminated. Now, another advantage of using biomodern is that when the dye is being extracted from the plant part of the dye yielding plant, at that point of time only these biomodern yielding plants also can be co-heated together. That means they can be boiled together, and thus the moderating and the dye extraction, or rather the extraction of the mordant and the dye extraction can be done simultaneously. Now, this extract already has the mordant now in the form of biomodern. So, if the extract is simply concentrated and dyeing is carried out, one full step will be avoided in the dyeing process. Other biomoderns, as I said, are gallnut, that is Majufal, Quercus infectoria, or myrobalan, which is Harda, which is also called botanically as Terminalia chebula. It also can be used in place. So, these are myrobalan and gallnut have been used from time and again, but nobody actually knows whether they have any metal content or not, but they definitely have tannin content. So, maybe the tannin is contributing. So, they can be also classified as biomoderns, and of course, we have tannins from various barks of the plants, and therefore, it is possible to actually use these tannins very easily for the purpose of dyeing. Now, tannic acid also, synthetic tannic acid can be also used at the same time. So, therefore, if we have to then look at various other parameters of dyeing, rate of dyeing, kinetic of dyeing, enthalpy of dyeing, entropy of dyeing, adsorption isotherm, dye affinity, dye uptake, all this can be ascertained. There are procedures available, but it is more from the scientific point of view that these parameters are tested for dyeing.